Alright guys, so Screencastify was not cooperating too well with me. Couldn't get the sound to work, so we're gonna basically just go ahead and do it like this. We're gonna start going through some of this chapter 15 stuff that I asked you guys to take a look at. So it's genes and how they work. Now obviously we have probably about 20,000 genes in our body, but about 100,000 different protein products. So how are we turning these genes on? Why do they get turned on? When do they get turned on? How do they get turned off? How do we regulate them? Because just because you have a gene doesn't mean you need to have it on all the time. So we're going to take a look at that. Now, a little background information that we got to take a look at is obviously the nature of genes. We know that genes are basically DNA. DNA is the inherited material that you get from mom and dad. One of the first guys to start taking a look at this was Garrett, and he looked at certain diseases being more prevalent in families. We obviously understand that from a genetic viewpoint. Recessive traits tend to show up more. So he was looking at a disease called alkaptonuria, which basically means that this family, they had black pea. And so he found that pretty much all of them had it. So there's that familial relation, passing genes or traits from one generation to the next. And it seems like everybody in this particular family had this gene turned on, so they were all having this black pea. As far as starting to make some connections between some things, Beetle and Tatum, these guys come along and they started to make the leap between DNA and enzymes and proteins and that type of thing. And they did this by looking at this Neurospora crassa experiments. And basically what this experiment says is that for every gene you get one certain polypeptide or one certain protein that you're going to manufacture. And basically what their experiment boils down to is that if you mutate certain genes, if you mutate one gene, it's not going to allow other things to turn on and work. So what they had to do is, what they found is that if you want to mutate gene E, then you can't turn on gene F and G and so on and so forth. So one gene basically leads to the next, to the next, to the next. And if you don't have gene E turned on, then you can't turn on gene F, you can't turn on gene G, and you can't turn on gene H, and you can't make your final end product that you really want. So pretty much every gene that we have is going to have one protein product that goes with it in some way, shape, or form. Central dogma, simple enough. We know that for both eukaryotes and prokaryotes, right, we're going to go ahead and we're going to have that template strand. We're going to go through the process of transcription of turning DNA into RNA and then RNA into our protein. We know the central dogma over here, replication, transcription, translation. There is one little weird piece here, this reverse transcription, which we'll end up talking about that a little bit. That's kind of a newer piece that's been added to the central dogma in recent years, and we'll see why going from RNA to DNA is not quite, you don't quite get the same thing. It'll make sense when we get to it. All right. So as far as how do we actually go ahead and make some RNA? Now we're going to try to make mRNA, that's our messenger RNA. So we're going to take our DNA, so here's our DNA, we've got our 5' and our 3' ends here, right? That anti-parallelism, okay? So what we're going to do is we're going to take this template strand right here, this 3' to 5', and that's the one we're going to read. And we're going to read that one because RNA polymerase, which is going to be the enzyme that we're going to use to make our mRNA, likes to work in the 5' to 3' direction. So as it builds, it's going to build a 5' and another 5, so it's going to read this way. So when we get down here to our coding, we're using this template or antisense strand. So A to U, G to C, T to A, so on and so forth. And that's how we're going to build this 5' to 3' mRNA strand. Okay, so the uncopied strand, that's our coding strand. The one that we're going to look at and we're actually going to use is going to be this 3' to 5' to build this strand down here. Okay, now we got to go into and do the translation process. Now transcription's easy. I mean, if you know to switch out a T for a U when you're going from DNA to RNA, you're fine. Transcription's easy. The translation is where we got some more stuff happening, and I know some of you guys are worried about some of these things in here. So let's take a look. There's all these different types of RNAs. There's mRNA, that's the messenger RNA. Now, that's going to be basically our recipe. That's going to tell the ribosomes, hey, here are your list of ingredients and where we need to put them together in. So we're going to need this thing to tell us, hey, here's what we're actually making. Now, our RNA, that's the ribosomal RNA, this is going to be kind of like the kitchen. This is where we're going to do the assembly. This is where we're going to put everything together. So we're going to go out into the cytoplasm, we're going to find a ribosome, The messenger RNA is going to go through the rRNA, and the rRNA is basically going to kind of read along and say, oh, get me this amino acid, get me this amino acid, get me this amino acid, and put them together in this order. Now, the person that's going to be our gopher and is going to bring us the ingredients that we need is going to be tRNA. So tRNA is literally going to bring us our amino acids. So we've got recipe, we've got place to cook, and we've got our ingredients essentially in the tRNA. Now, there are some other things we could really get into. I'm not going to worry about it. If you really want to, get into some more of the small nuclear RNAs or the signal recognition particles, you are more than welcome to really get into that. I'm going to live over here and focus over here because I think that's the most important stuff. I just threw that in for extra. All right. Now, here's the deal. When we're actually reading, right, and we're reading the genetic code, we have to read it in chunks of 3, and that's going to be our codon. Okay, and this is what's going to be called our reading frame. As you can see, depending on how you space things out or how you chunk things out, and I think some of you guys figured this out last week on that one little problem I gave you, like on Friday I think it was, for the warm-ups. If you have to move over a couple spots, it's going to change the way you read the whole sentence. So here we go, we delete one letter. Why did the red bat eat the fat rat? Why did he? I mean, just by changing that, shifting that reading frame over one spot, it's going to be a little bit different. Down here, they deleted one letter, right? So they take that T out again, and why did her ate at, and then it makes no sense. So depending on where your reading frame is and where you're looking at that start codon and moving along, it's going to have an effect on the end product. Okay, so these are just different genetic codes. Sometimes you see the circular ones like this. On the circular ones, you start at the middle and work your way out. So this would be like first letter, second letter, and then third letter. So A, U, G, and there would be methionine. I tend to use the square ones. Here's AUG, methionine, that's your start codon. And then over here you've got your 3 stop codons, UAA, UAG, and UGA, and they do exactly what they say. They tell you to stop. So I think you can all read that pretty easily. Obviously, UAA, UGA, and UAG stop codons. AUG, always the start codon. Oh, I got a typo in my notes. That should be not fro-methionine, it should be for-methionine. So, obviously, does that mean then that every single protein that you're going to manufacture will always begin with methionine? No. We'll get into some of the stuff that's going on a little bit later on with that. But not every single end product enzyme or protein will start with methionine. Okay? Some codons can code for more than one amino acid. I think you can see that if I back this up just a second. You could see up here, like there's phenylalanine. There's 2 different ways to ask for phenylalanine. If you look over here, there's 4 different ways to ask for glycine. So basically, it's what's known as a degenerate code. Basically, the first 2 letters are really important, and that last letter is kind of that wobble spot. But there's more than one way to ask for a lot of these different amino acids. Code's nearly universal, so I don't care what kind of living organism you're looking at. Morty's laying here on the sofa while I'm making this video. He's got the same DNA as I have. We got the same DNA as like the grass out back. The only differences tend to be in mitochondrial and chloroplast DNA. They seem to be slightly different, but we're going to focus more on just the regular DNA found in a nucleus anyway. So, all right. We're going to go through this little chunk right here. I know we're probably going to call it quits for this video. This will be enough for you to take a look at and kind of soak in and maybe think about for today. But we're going to talk about prokaryotes first. Now, prokaryotes, obviously bacteria, very simple. This is the prokaryotic RNA polymerase complex. Now, as you can see, it's made up of 5 different subunits of proteins. So again, this would be a quaternary structure. It's made out of 5 subunits. It's got 2 alphas, it's got 2 betas, and then it's got this little like sigma subunit down here. Now the way this works is it works kind of like pinchers, like a fingers, right? So what's going to happen is that this subunit right here, these things are perfectly spaced out so that as they come up to the DNA in a prokaryotic cell, they land at certain sites. So we've got this promoter sequence, TATAAT, right? And then we've got this other promoter sequence. And these fingers, these little pinchers here, are perfectly spaced out to basically kind of read along the DNA, find those little areas, and then grab onto it. What they do is once they find those areas, these start sites, you can see it kind of pinches. And as it pinches, it's going to kind of lock this RNA polymerase in, right? We're going to get our little transcription bubble because we have to open up the DNA, just like we did in replication with like a replication fork. So we got to open up this little transcription bubble, okay? We're going to have our sigma subunit is going to get out of there, and we're going to start the RNA synthesis. So what's going to happen now is this is going to read along. It's going to read along that template strand, and it's basically going to go ahead and make us some mRNA. So if it sees an A, it's going to put a U. If it sees a C, a G, and so on and so forth. Okay? Now, it's going to keep going and going and going, and as you can see, it's going to read along. It's going to read this little template strand and just keep adding things and unwinding and closing things back up. But at a certain point, we're going to get near the end of the recipe in our DNA gene, right? And what happens is the RNA folds back on itself, and it does this little hairpin loop. And when it does this hairpin loop, it basically causes this to fall off of the DNA and dissociates. Our RNA polymerase is going to dissociate from the DNA, and everything kind of separates. Now, what's going to happen to the mRNA? Well, we're in prokaryote, so it's already just floating around kind of like a nucleoid region. It's going to have to then go and basically bump into a ribosome, and it's going to get turned into a protein. Okay, and I think that's where I'm going to stop for right now. I'm going to save the eukaryote transcription for later, and we'll see how that goes. All right, thanks.